Right now, there is a microscopic anomaly that breaks the standard rules of human cellular biology to exert control over the host. It operates through a chemical language that our own DNA cannot speak. In the intensive care unit, this control manifests as a severe clinical crisis. Patients present with high-volume intestinal paralysis, toxic megacolon, and suspected systemic sepsis, all driven by a total failure of gastrointestinal motility. The agent driving this paralysis does not exist in a single strand of human DNA. Human cells completely lack the biological machinery to manufacture it, yet it is currently dictating the patient's survival. This molecule is pyrrolysine. It is the elusive 22nd proteogenic amino acid, a structure that functions outside the standard 20 amino acid biological alphabet. Because pyrrolysine falls outside native human metabolism, the primary driver of this patient's critical condition remains completely invisible to standard laboratory panels. To understand this anomaly, we have to look at the UAG amber codon. In humans, this genetic sequence functions strictly as a stop signal, telling the cell to terminate the protein chain. This schematic shows a human ribosome tracking mRNA. Hitting the UAG sequence, it recognizes the stop signal and releases the protein. But commensal archaea read that exact sequence as an insertion command, immediately adding a specialized molecular block instead of stopping. That block is pyrrolysine. Structurally, it consists of an L-lysine core framework where the terminal nitrogen is linked via an amide bond directly to a specialized ring system. This is a blueprint of the pyrrolysine molecule. The terminal ring, specifically a 4-methylpyrroline 5-carboxylate, functions as a highly localized electrophilic trap. These ancient microbes utilize a software-like macro execution to hijack a universal stop sign, allowing them to build a highly active metabolic trap within their proteins. This genetic divergence creates the absolute zero rule. Human vascular, visceral, and skeletal systems contain strictly zero endogenous or free pyrrolysine. This diagram maps the geographic boundary of the molecule. While the rest of the body is natively blind to it, pyrrolysine is sequestered within the cell walls of commensal archaea residing in the colonic lumen. Synthesizing this molecule requires a heavy thermodynamic tax. These archaea must expend significant ATP and NADPH to build the ring structure in an environment where nutrients are already scarce. They pay this price to achieve niche monopolization. When we digest foods like eggs or beef, choline breaks down into volatile nitrogen-dense methylamines. For standard gut bacteria, these methylamines are metabolic dead ends. The pyrrolysine electrophilic trap captures these methylamines, lowering the activation energy required to convert them into methane gas. The microbes endure this massive metabolic tax because it guarantees them an exclusive energy source derived from our dietary exhaust. In a healthy state, this process is balanced. But in the ICU, standard medical interventions can trigger a clinical failure known as intestinal methanogen overgrowth, or IMO. High-dose broad-spectrum antibiotics eradicate the bacterial pathogens, but leave archaea untouched. This creates a state of unmonitored proliferation. Archaea survive because their cell walls are composed of pseudopeptidoglycan, which lacks the binding targets targeted by traditional antibiotics. This competitive release causes UAG translation to go into overdrive. As the archaeal population spikes, local methane production reaches critical levels. This diagram illustrates the mechanical consequences. Methane gas diffuses into the intestinal wall and hyperpolarizes the interstitial cells of Cajal, the pacemaker nodes of the gut. This flatlines the rhythmic peristaltic wave. The resulting stagnation leads to toxic megacolon and mucosal barrier failure, creating a severe risk for bacterial translocation into the bloodstream. IMO represents a direct methane-induced neuromuscular paralysis of the digestive tract. The most frequent diagnostic error occurs when clinicians order a standard plasma amino acid profile to hunt for pyrrolysine. Because of the absolute zero rule, the analyzer will return a blank result every time. Pyrrolysine does not utilize human transport systems to enter the blood. Diagnosing this pathway requires sequence-specific metagenomic qCPR of stool specimens to quantify the archaeal PYLRS gene, the genetic machinery that builds the molecule. However, this approach is vulnerable to the microbial DNA degradation trap. If the stool sample's cold chain is broken, the results become meaningless. At room temperature, the archaeal cell walls undergo nonspecific lysis. Ambient nuclease enzymes swarm the exposed DNA, rapidly shredding the Pyrrolysine target sequence and triggering a false negative result. This analytical failure provides dangerous clinical assurance, while active paralysis continues in the patient's gut. The gold standard for a confirmed diagnosis requires a cross-check between the genetic footprint and functional output. This flowchart tracks the proper workflow. An elevated stool Pyrrolysine gene count must be paired with a methane breath test showing a peak concentration over 10 parts per million for a confirmed diagnosis. Accuracy depends on strict QA/QC, including calibration against international reference plasmids and enforcing a total allowable error of plus or minus 20%. Treatment requires a 14-day protocol, combining rifaximin and neomycin to dismantle the overgrowth. Neomycin is the critical agent. It selectively binds to archaeal ribosomal subunits and destabilizes their pseudo-peptidoglycan walls. Precision antibiotic targeting is the only way to dismantle an organism operating on an entirely orthogonal cellular architecture. While gastroenterologists use neomycin to eradicate this molecular machinery, clinical bioengineers are actively harvesting it for the opposite purpose. Pyrrolysine serves as the perfect orthogonal biotechnology signal node. Because human cells cannot process it natively, the system operates with zero biological interference inside human cell lines. Researchers isolate the PyLRS enzyme, alter its binding pocket through targeted mutagenesis, and transfect it into human cells. These transfected cells now read the UAG stop codon as an insertion command, exactly like the gut archaea. As shown here, the ribosome grabs a synthetic, non-natural amino acid and precisely inserts it into the framework of a targeted monoclonal antibody for advanced cancer therapy. We are utilizing an ancient microbial framework as a microscopic manufacturing plant for next-generation pharmacology. This provides our first two clinical pearls. First, pyrrolysine is a somatic blind spot. Never use serum panels for detection. Second, an overgrowth is a mechanical neuromuscular pathology where methane hyperpolarizes the enteric pacemakers. Third, a breath test exceeding 30 parts per million indicates a high risk for toxic megacolon. Fourth, treating the archaeal source requires the specific addition of neomycin. Finally, remember that a broken cold chain causes DNA degradation, which is the primary driver of false negative diagnoses. These are the 5 architectural pillars: UAG hijacking, electrophilic trap methanogenesis, absolute zero compartmentalization, neuromuscular paralysis, and orthogonal bioengineering. The distinction between human cell biology and microbial engineering has blurred. The molecular tools currently digesting your lunch are providing the mechanical architecture for targeted cancer therapies.